So welcome to everybody who's coming into the room right now to our first CCMA 2021 Shared Resource Labs Core Facilities Perspectives series. How many people do we have? We have 39 right now. We're just a little bit early. So I think I'll wait till we are on the hour before we start. We're still waiting for Josh, I think.
I just typed Josh a message, but I didn't get any response yet. So.
I think he's in a meeting right before this one. So he is going to be a co-moderator with myself. And, the slides that you're seeing right now are from our sponsors. So we have BD Biosciences as our series sponsor. And for this particular session, we have Apogee Flow Systems, Beckman Coulter Life Sciences, Biotechny, Cytech, as well as Luminex. So really thankful and grateful that they have contributed and sponsored our event. And as we were speaking about before, the proceeds to the sponsorship or from the sponsorship is going straight to a new travel award named after Christopher Spring, who was our past president. We miss him very much, and I believe this initiative would be very much approved by Chris. So the idea is that the award will be given to core managers across Canada to come attend the next in-person CCMA. Lots of familiar names in the list, so really happy to see Canadian Flow Cytometry making a show. It's really nice to see everybody, be able to connect. Miss talking to you all. Okay. Dr. Welsh, are you showing up? Okay. Couple more minutes and I think we're going to start. It's always surprising the number of registrations you get to the number of people that show up to the meeting. But it's nice that we have a bigger room now.
Is this going to be recorded, Vera?
It is. It is recording right now, actually, but we'll probably trim off all of this stuff at the beginning. So for those of you just joining us, it is noon. So I believe we should probably start. All right. Oh, wow. Okay, here's everybody. Hello, everybody. How many familiar names we're seeing from across Canada. Really nice to see you guys all. It's too bad we haven't been able to have another in-person CCMA in a while, but hoping that's coming up soon. And for those of you who caught the slides beforehand, they were from our sponsors for this event. We have a series sponsor, which is BD Biosciences, as well as individual sponsor for today's event, Apogee Flow Systems, Beckman Coulter Life Sciences, we have Biotechny, we have Cytech, and we also have Luminex, all of these great people who have made it possible for us to fund a new travel award, which we have named after Christopher Spring, who is our past president, who is no longer with us. So in the next in-person meeting, we will be featuring this session and all the other sessions, core facilities from across Canada. If you would like to be featured, please send us a note. And what that means is if you're featured in our Meet the Cores of Canada, it means you're going to be able to apply for this travel scholarship, which is specifically for members of core facilities. So without further ado, I think we should hop in. And what I'd like to do is for all of our panelists, as well as our guest moderator, to give a short introduction, who they are, what kind of small particle flow cytometry experiments they do, and what type of instruments they use to do these studies. Still got a lot of people joining. So do we want to start with Joanne, my right hand?
Okay, I will start. So my name is Joanne Lanigan. Currently, I'm the CEO of Cytometry Service, which is a consulting company. But as many of you know, I have many years experience running a core facility at the University of Virginia. And so I understand your trials, your tribulations, and the things that you try to achieve with excellence. So that's appreciated. As far as small particles, I started about 10 years ago collaborating with a group at University of Virginia who primarily works with plasma and EVs. I continue to collaborate with them. I initially worked a lot with the imaging flow cytometer, the image stream, and recently I'm working with the Cytec Aurora. Did I answer all your questions?
You did. You did a great job. So Des, you want to go next?
Hey, my name is Desmond Pink. I'm the Chief Science Officer for Nanostics. We're trying to develop and commercialize a blood test that would help men that may have prostate cancer not have as many biopsies by using small particle analysis by flow cytometry. We're currently using the Apogee GXP. In the lab, we have some A60s and an A50. And we run primarily plasma, serum, urine, a little bit of cell culture, and as many beads as I can get my hands on for troubleshooting and other types of analysis.
Okay, Edwin?
My name is Edwin van der Pol. I'm with the Amsterdam University Medical Center in Amsterdam, the Netherlands, as a scientist. And for one day in the week, I'm a Chief Scientific Officer of Exometry. I have a background in applied physics and did my PhD on the detection of vesicles. Now, by training, I have no training in flow cytometry, but yeah, after my PhD, I gained some experience. That was with the BD fax caliber, which basically caught fire during my PhD when I was working on swarm detection and light scattering theory to understand the limitations of vesicle flow cytometry. I led an international study to standardize vesicle concentration measurements that involved 64 flow cytometers, and now we are using an Apogee A60 micro and a customized BD fax canto. And for the rest, we are developing two entirely new flow-based instruments.
And John Nolan?
Hi there, I'm John Nolan. I'm a professor at the Scintillon Institute, where my group develops new tools for cell and molecular analysis. When I was working on my PhD, I was actually working on lipid vesicles and membranes. So sort of at the dawn of the molecular biology age. And not only did I not study DNA or proteins, I didn't do any blots or gels either. I instead used a flow cytometer, which put me sort of on the outskirts of science for a time. And it took me some time to catch up to get with all the genomic trends that were so in vogue. And so it's kind of interesting that, some years later, flow cytometry of membrane vesicles turns out to be important. So we're interested in vesicles, but there's also other sorts of nanoparticles. All nanoparticles are not the same. Vesicles are very different than other sorts of nanoparticles, so that's important to keep in mind. In terms of instrumentation, the last instrument I truly knew how to run was also the fax caliber. It never caught fire on me, but was wholly inadequate to measure vesicles. So our first instrument was a home-built instrument that was we built in order to have the sensitivity necessary to measure dim particles. But more recently, we've been working with some of the newer generation of high sensitivity instruments, the Beckman Cytoflex, the Amnis, now Luminex image stream and cell stream. I'm also CEO of Cellarcus Technologies, which is commercializing some of the developments from my academic lab.
So thanks, John. And now we have Josh, who's also a co-moderator with myself. And after Josh, I screwed up. I am going to let Camille introduce her core facility because she's part of the Meet the Core Facilities of Canada.
So I'm Joshua Welsh. I'm at the National Cancer Institute, where we're using EVs as translational biomarkers. I did my PhD in Southampton, and I developed with Thermo Fisher Scientific a high sensitivity flow cytometer. So I spent my PhD taking apart a flow cytometer and trying to put it all back together again. And I also had experience there with the Faxaria, the Cantos, and the Fortessa. And since moving to NIH, we now use the Cytec Aurora, a slightly modified version, and a Cytoflex, as well as a Astrios and a Nano FCM. So we play with a lot of flow cytometers. And one of the main things that I've been trying to help contribute a software called FS, which calibrates light scatter and fluorescent signals so that we can start talking in standard units, which I guess will be a topic of today.
All right, so before we dive into the small stuff, we're going to hear a little bit from Camille.
Yes, hello everyone, and thank you to the CCMA for the opportunity to talk a little bit about our course, or like, I guess I was the first one to register, so I'm the first one to start. I work at the McGill Flow Cytometry core facility. Our facility exists in its current form since 2018. Before that, there used to be 2 core facilities at the downtown campus. I was at the microimmuno department core. My colleague Julienne Lecomte was at the cancer center. And we both thought it was absolutely ridiculous to have 2 competing cores within 300 meters of one another. So we luckily managed to convince our administrators that we would be stronger and better working together. The fact that I was working in a building that was almost crumbling and falling apart probably helped the push. I see Marie-Hélène acknowledging because, yeah, she's been there before my time. And so since 2018, our newly merged facility is co-managed by me and my colleague Julien. We have an oversight committee of 5 faculty members that kind of represent our main users. So we have people from immunology, from cancer research, being the flow core on the downtown campus means that we need to service pretty much all of our departments. So it means that we need to be good and not expert, but at least know some stuff about various type of cells and particles. We'll have people from biomedical engineering coming and just working with beads coated with DNA or things like that. McGill has a School of Agriculture and Animal Studies. We have a farm on the West Island of Montreal. So we'll have sometimes to do panels for chicken lymphocytes. I've had to sort bovine samples at some point. We have a lab up north at the Arctic, so we sometimes have soil samples from the Arctic that we need to sort or analyze. So we really, really love weird stuff, and we tend to have like lots of unusual or untypical requests coming our ways, and we absolutely love it. We currently have 5 cell analyzers and 2 cell sorters in our facility. So we have 4 BD cell analyzers going from 2 lasers to 5, so 6 colors to 18 colors. we were very happy to purchase an Aurora right before Christmas, so we haven't had much time to play with it yet. But
are you guys going to be doing small particles on the Aurora?
Absolutely. So the lab right next door from us is a gut microbiology lab, and when we think about bacteria, bacteriophages are just as present as bacteria in the gut, and we want to use the Aurora to kind of study a little bit more all those bacteriophages in a more pleasant way than having to count them under a fluorescent microscope, because that's apparently very, very long. We're also
that's awesome. I think one of the nice things about being connected to the other cores around you is that you'd be able to share in your expertise and your experiences. And so I think that's definitely one of the goals of this series, is to connect people, to meet people, the fact that we can't meet people in person. But yeah, so thanks so much, Camille, for sharing the details about your core. And yeah, and so hopefully you and Julian will be able to join us at the next CCMA, and we'll be able to help you along with that.
Oh yeah, absolutely.
Okay, so I mean, I think this is a new experience for everybody. I think the panelists all know each other, and we've all had discussions with each other about EVs. So the idea is, how do we do this in an organized fashion that other people can understand what we're talking about? So Josh, do you want to sort of start off on kind of like broad topics that we want to cover today, or?
Yeah, so I guess the important thing is it's not a debate, it's just a discussion.
Yeah.
So I guess there are key kind of things that keep coming up in the EV field. And I guess today we wanted to kind of have a discussion about them. And the three main topics are, do we use a fluorescence or a light scatter trigger? It's a question that often comes up. Calibration is a word that's often used and comes up, but it's implemented in different ways, and it's a useful thing to discuss. And also about field progression and development. And we keep talking as well about moving on to more and more sensitive equipment to look at EVs, and what we don't often think about is the implications of actually moving to more sensitive EVs, and all of the other stuff that's already a hassle will be extra hassle to deal with when we get to these platforms. So I guess to start us off with fluorescence and light scatter. So would any of you say that you exclusively should only use a single trigger, or do you think that it's a subjective thing? I guess we should just jump in and kind of have it more of a discussion than.
Yeah, I think a quick and easy answer to that is very application dependent, right? It really depends on what your end goals are. It depends on the type of samples and whether or not you have a good pan fluorescent marker that you can trigger on fluorescence. I mean, if you can have that's I've seen, right, triggering on fluorescence, but that's not always the case in every application. I think whichever approach you need to understand the nuances are for each, and what the considerations you need to make with regard to calibration, especially. I'm sure we'll get into that as well. But I really do think it's application dependent.
Yeah, I agree with Joanne. I think it comes down to application, but if you make a decision one way or the other, depending on the application, you have to be able to defend it. So you need to do the baseline experiments to actually show this is behaving, my trigger is behaving the way that I expect it to behave. I think that's what it comes down to. Whatever you choose, you have to be able to defend it.
And guys have.
So I'll tell you what I think. So I guess it depends to some extent on whether you are developing your own assay or you're using someone else's already developed assay, in which case, you'll use the other person's assay. And I think this, is a good time to bring up the point that there's a difference between instruments and assays, right? There's a lot of discussion about which instrument is best. But you have to remember that the instrument is only a part of the assay. It's an important part of the assay, but it's, you know, one could argue a small part. Even more important in most applications, in my experience, are the details of the assay, the reagents you used and how you use them, how you operate the flow cytometer, and then how you analyze the data after you've collected it. And so you can't really talk about any of these issues in isolation. They have to be talked about in the context of the assay that you're using.
Yeah, all right. I totally agree that yeah, the trigger detector is instrument and assay dependent. I think in the literature you find more claims that generally the fluorescence detector is more sensitive. Yeah, a colleague of mine showed that that's not applicable to any flow cytometer. And then I made a plot which I would really like to share with you. And that's this one. Because, oh, can I make it full screen? Yeah. Because I realized that in case you trigger on fluorescence, yeah, the smallest detectable diameter of vesicles highly depends on the label you are using and the number of receptors, or basically the receptor density on the vesicles. And yeah, I think we need to be aware of that in case we trigger on fluorescence, that the detection limit is not only determined by the instrument, but also highly by the label used and the receptor density. It's of course the same with scatter, but the more sensitive the instrument is, the more variations in the refractive index, which you see in the right plot, and the more they, yeah, how to say, have a trend to come towards each other. And even though vesicles have different refractive indices, that doesn't affect the limit of detection that much compared to the labels I have selected here to make the plot on the left. And in this case, this was CD41 platelet marker, CD62P also activated platelet marker. And in this case, arbitrarily selected IgG control.
So I think, yeah, I was going to say that I think whichever approach you take, I think Edwin's point of limit of detection is important, right? That's why calibration is important, so that we know from reported data what the limit of detection you're talking about, whether it's an MESF or in meters, because that's really important. Both are going to give you different, or potentially could give you different populations. You can have the sample, go on scatter and get one set of results, or you can take that same sample and trigger on a pan-fluorescent marker and get a totally different set of results. So I think it's important to, whichever approach you take, to make sure that the data is described in terms of the limit of detection that you've established for both approaches or either approach.
But back to the issue of instruments and we can understand that we can characterize the instrument performance by itself, right? We can talk about its background and limit of detection in units of photons, photoelectrons, or MESFs, which are intrinsic properties of the instrument. And then there are the properties of the assay that you apply to that instrument. And you know, those aren't the same measurements. They're different steps, and it's useful to consider them as separate objectives. So yeah, there's instrument performance, and then there's assay performance.
Yeah, I mean, to agree with John there, I think one of the biggest things, and I think the NIH has brought this up in any other major institute, antibody variability, shipment to shipment, batch to batch. The quality of your antibody will help define your LOD, and you know, controlling and validating your antibodies every single shipment that you get is critical, absolutely critical. And if you think that you're getting the same antibody from one shipment to the next, even if it's the same lot number, you really still need to test. These antibodies may be generated, say on January 1st, 2020, they may be held in storage until 2022, packaged in 2021. So, you know, what happens during storage, we don't know. So each shipment you need to validate, because of course that will all trickle down to your LODs.
So Duncan, you build that into your assay workflow. And if you were to publish on your assay, you would include that information so that others who wanted to adapt your assay could do it.
Yeah, we titrate. The amount of titrations that I do is almost scandalous. And then it's the titration of each antibody, but then we do crossovers with known control. So before he goes into the clinical test, there are crossovers done to make sure that the data matches. And that goes back with a Livy Jennings assay.
So this is probably a good time to promote the guidelines that have been recently published by ICEV that sort of lay out, what you need to report about your assay, right? If lot-specific characterization of antibodies is critical to your assay, you need to include that so that people know that they do that as part of their assay. And if you're developing a new assay, based on principles, that are new, likewise, you have to look out for things like that and report them.
Yeah, because I think it's critical for people to appreciate that, once you receive an antibody or a probe and there's an expiration on the label, that expiration date is not necessarily based on stability data. That is based on it's 1 year from your receipt of product. So it's out of the vendor's control, and they give it a sort of a statutory 1 year. It's not necessarily based on any stability data. And if you want to confirm or actually talk about stability, you actually have to do those assays.
So that maybe, go ahead.
Something you said, John, about if you're using someone else's assay and just applying it to your instrument, I think it's also important to understand that just because someone's published an assay on the same instrument you have, it doesn't mean it's the best assay for your instrument. We still have a lot of variability between alignment and what the most sensitive channels are. And I guess that ties into what Desmond says, where we really do need to defend why we picked our settings and an assay, regardless of if it's published or not, and regardless of the same instrument. We can't just point to said so.
Yeah.
Right. The rule of thumb in published academic research is that you can believe about 15% of it. The numbers probably lower in this field, right? So, reader beware.
Yeah.
Well, I think the other important fact to bring into with regard to reagents is that certain reagents with certain types of vesicles may react one way, where completely different vesicles with different lipid composition may behave totally different, right? Or especially when you're dealing with lipid membrane dyes.
Yes.
So people need to be aware that, because they read a paper and somebody used this lipid membrane dye and it worked well with their assay, with their types of EVs, it doesn't necessarily equate to all EVs.
So this might be a difficult question to answer, but in context of people who are just approaching EV flow first time, what would you suggest? Like, how would you approach this kind of difficult field where there's no standards, where instruments, when we're using them at levels where they're all kind of not the same? Because when we're running cells, we're assuming a certain level of performance, and I think that's what gets us into trouble sometimes, is that some of us are so familiar with the technology, but we're not familiar with it at the level of EVs. So as the panelists, what would you suggest is the best way to approach as a newbie EV flow cytometrist?
Pick up the phone and call someone. I'm serious.
Are you giving your number?
I work with a lot of different people. I mean, I call because there are times when I'll find stuff and it's like, I have absolutely no idea. This is brand new. I've got a different type of sample, or I'm trying this dye for the first time. Just pick up the phone and call someone.
I think there's also a lot of really good literature out there now. And I think, there's members in this panel right now who've actually contributed quite a lot to a lot of the literature out there. I mean, I guess this is the age of watching videos, and there are actually a lot of videos out there as well from members in this panel. But some resources, I think. MyFlowSite EV is a great one. I think there's a lot of associations that are putting out literature as a start. Any comments from panelists?
Yeah. So there is this EV flow cytometry working group, which contains members from different societies. And indeed, last year we published MyFlowSite EV reporting framework. It's a framework that recommends you what to report. It doesn't explain the principles behind it, but it may refresh your mind because if you are triggered of what to report, you may be triggered to do certain controls. Now, to explain all the details behind that, it's a lot of work. We are drafting currently an educational manuscript. Right now, it's 30,000 words and almost finished. So keep an eye on that. It will be rather a compendium, not something which you read in 1 hour. But we hope it's going to be helpful because then a lot of literature which has been published over the recent years will be bound in one compendium.
So the biggest limitation for people getting started is lack of good reference materials. And, I think if we had that would be a good place for people to start so they can try different things, try scatter versus fluorescence. And, you have to have some kind of known to some extent where you have, what to expect in terms of fluorescence or concentration or scatter properties. I think this is what the field lacks right now and is making it very difficult for people because starting with a sample that's just a total unknown is very challenging. So I think we have a ways to go with getting some good reference materials out there that people can start to establish things like instrument settings and sensitivity and so on with some kind of relative known sample.
So do you not think that the available kind of reference materials and calibration that's available can determine the optimal instrument settings? Are you talking more about the validation with EVs and an assay, or are you talking about the instrument itself?
So if I'm talking about a starting place for people to try and figure out where to start, is it going to give them the absolute end all be all instrument settings for their particular instrument? No. I think you can share more details on the better way to do that. But I think for most people, as Vera was asking, where do people start? This is at least a place to start to figure out whether or not your instrument is even capable of detecting some reference material at all, regardless of whether the setting is optimal, right? So, and to see what the differences are when you trigger off a scatter versus fluorescence and how that affects your results. There's technical information that is needed to determine optimal in settings. That is probably the better way to do it, but at least this would get people started somewhere, or better than that, where they are now, trying to within a totally unknown.
Yes, I think. Go ahead.
Okay, so I'll take issue with Vera's statement that there are no standards and where should one start, and this will be, I'll try to keep this commercial
it's Joanne
free, right? So I'm conflicted because we've developed an assay.
Don't blame Vera. It was me, John.
So in my lab we've developed an assay based on fluorogenic membrane probes that can selectively detect size and immunophenotype EVs, and this is the vesicle flow cytometry kit from Cellarcus. And so my suggestion would be, if someone's just getting started, try one of the try a kit that's developed and optimized for this purpose.
Does it work on every instrument though? That's the problem.
Well, I mean, so the kit provides standards in the form of intensity beads, antibody capture beads, and reference vesicle preparations that are incorporated into the assay. So this is a complete assay. You think an ELISA, right? So you get a box of tubes, you get a protocol with a step-by-step protocol, you get a analysis template that leads you through the data analysis, and you get a result, right? So this is one solution to the vesicle problem. It works on instruments with suitably sensitive fluorescence detection. And for us right now, that includes the Cytoflex and the cell stream and image stream from. We're working our way down the pecking order of fluorescence sensitivity because we use fluorescence to detect the vesicles. So the instrument must have the ability to detect a particle based on dim fluorescence. And you know, the number that meet that criteria is growing all the time. I think there is an awareness in the industry that you can't simply rebrand a lymphocyte analyzer and call it a vesicle analyzer, that there actually needs to be some rationale behind the instrument design that enables detection of dim particles. So we haven't seen the end of the instrument evolution. Thinking about what kind of assays are going to be able to be performed on different instruments, you know, is it also an important part of the question. So there are commercially available standards that are relevant to flow cytometers. And getting to Josh's point, yeah, step one for us is ascertaining the performance features of the flow cytometer to sort of make some judgment about whether it's going to be possible to measure these dim particles, and then to actually have some dim particles that serve as reference materials or positive controls in the assay to confirm that you can measure them. And I think that's a rational way to think about any assay development. You know, if you want to develop your own new assay in your own lab, you probably want to decouple instrument performance and assay performance, and then document and validate those in an appropriate way.
So from what you've said, John, I think is a perfect segue to calibration. So the first step, in my opinion, is to find, you know, if you go into a core, which instrument do I even start using before I want to run these? And the only way we can do that is if we're able to look at our analysis on the same metrics, not on these arbitrary units, but a common metric. So the first thing that I think would be really great to hear from all of you is what calibration, the definition of calibration means to you. Is it's thrown around, standardization and calibration is thrown around hugely in the EV literature, and it can mean anything from just analyzing some beads and saying the scale was calibrated because I can detect 200 nanometer polystyrene, all the way to doing regressions and me theory. So I guess, Edwin, you'd probably be a good person to start because you've spent a lot of time in the metrology kind of area.
Yeah, so in my opinion, yeah, just running your daily beats is not a calibration. Yeah, the idea of a calibration is that you go from the arbitrary units of measurements to standardized comparable measurement units, preferably SI units. This is not easy, especially not for flow cytometry. I think flow cytometry has a long history of dealing with arbitrary units. And people get used to the plots they see. So at a certain moment, they don't perhaps have the tendency to do the calibration. But if we all calibrate our arbitrary unit scales in terms of vesicle diameter in nanometers, fluorescence in MESF, we express the concentration of vesicles we measure as the number of vesicles with a certain marker and an intensity and diameter within a specific range, we can start comparing data. And I think that's a prerequisite for that.
There's that part, but there's also the day-to-day calibration. So for us, we want to make sure, like, we're in the middle of a clinical study. So I want to make sure that my instruments are reading the same today as they go. So we run every single day, and we follow that up after calibration with monitoring beads. So we set all of our PMT detectors and our light scatter detectors based on our original settings from about a year ago, year and a half ago. And I can track to show that I'm getting the same readings daily, plus or minus a certain percent, of course. And you have to set that right at the beginning to say, this is what I want. And that's one of the hardest things, because even if new advances come along and you want to get more sensitive, you're set from day one and you don't want to change those criteria.
So Des, do you have a sample that you run every day that you establish some criteria for, that you look for the reproducibility of that sample over time?
So we've worked with Apogee on a set of calibration beads for size and color. And then I actually run the, the canonical Apogee bead mix. But I've done, I don't know how many lots of those, and exhausted the lots. And I can show you longitudinal data to get flat lines from the cytometer over 2 years.
I guess what I was asking was EV sample.
Those are the first set of calibrations we do, and we test our water, sort of our sheath, and our diluent as well to make sure that they're clean. But for each antibody, we've actually developed both a negative and a positive control. That is by far the most stressful thing that we've done, because it's so dependent. You have to make sure that when you're doing your stabilities, you need to have a reference material, where is your antibody stable, and how stable is your control. So it's a catch-22 at the beginning. But yes, we have developed controls for the negative and positive functionality of each antibody, and those are run before samples are.
So it's worth, I think, probably discussing that, going back to the application-specific nature of some of these things. So Des is he's at the forefront of applying EV analysis, in a regulated environment towards an actual diagnostic. And that's where I think a lot of people would go, although not all. But, that's a unique animal, really, that's a little bit different than what a core lab and a research university might encounter, where they might encounter lots of different applications, lots of different types of questions, as opposed to development of a fit for purpose assay for a particular diagnostic. I don't know, Des, you want to talk a little bit about that?
That's true, but all of the fundamentals are the same. Mm-hmm. So each day I start off and I ask the question, is my machine calibrated to the point where I want it to be, its original point? Are my monitoring beads showing the same, result? Is my sheath clean? Is my diluent clean? Are my antibodies working? So, one of the hardest things that I think anybody developing a new assay will be finding the right clone for your antibody. You may search a whole bunch of clones and they may work well on cells, but you may just find that they don't work well on EVs. And your workup in cell culture may not translate over to plasma or urine or serum. That's one of the harshest realities I think a lot of people find.
So one of the things you mentioned, Des, is you've used the word calibration a lot, and you've said running beads. But are the outputs of running these beads in standard units, or are they just running the beads and you have an internal, gates where they should?
So we have internal gates. If I need to translate it to standard units, I can. Our secondary analysis is quite unique. For the assay data, it's all done by machine learning, so we don't necessarily give it out in standard units. So
do you think it's fair to call it a calibration, or is it more of a quality control that
the machine is calibrated to a set point that was standardized at the very beginning.
So what are the units of that calibration?
So that unit, you could actually put it as MESF if you wanted to, and I can do that. I don't do it on a day-to-day basis. Or you could keep it into the standard. Sorry, go ahead.
I think to me, it sounds more like a validation or verification and not much a calibration. Calibrations are usually against a known traceable standard that has some known units associated with it. I think what you're doing is what we do in the clinical labs, even for cells, right? It's validation.
It's validation.
So
that's true. Desmond is at the stage because due to his comprehensive QC, that in fact, with a single measurement at the same settings, he can calibrate the axis if he wants.
Yeah, that's the thing. I can go back
which I
and
recommend, which I recommend to do in order to. I mean, if you want to scale up, for example, with different labs within your company, I mean, maybe a new flow cytometers in the future, which have to measure approximately the same as with your apogee. Or if you want other scientists to reproduce a part of your data, which becomes important, I think, at that point of publication, I highly recommend to do.
Yeah, I just wanted to clear that up, because you in your wording, Edwin, you said you could perform the calibration if you wanted to, and it's just the distinction between the two. So I completely agree. What Des is doing is incredibly meticulous, and he's getting very reproducible data. But for the field to learn, because we keep seeing in the field that people are saying they're calibrating and nothing's in standard units. So if someone else wanted to go and replicate the data, they don't have all of the standards on Des's instrument. They don't have Des's instrument. And so to report, we need to calibrate.
Yeah.
We need
for the field. For the field, this is essential.
Yeah.
Can I show a slide?
Sure.
This is just some thoughts about calibration. Can you see this? So this is, my simple way of thinking about what we're trying to measure. Flow cytometry has the, root of measurement in its name. And at the end of the day, we can expect to measure certain things. And what are the things we would like to measure about an EV? And I just lay out the ones that I can come up with here. We want to measure the concentration, the number or concentration. We'd like to measure the size, because that's a distinguishing feature of these things. And then we want to measure cargo, what they're carrying. I add on refractive index there as a potentially interesting physical property of the particle. And then when we have what the instrument can actually measure. And the instrument, actually can't measure any of those things, right? The instrument can count some stuff, and it can report arbitrary measurements, which, the base level is detected. If we want to get over here all the way to the right to these quantities that we want to measure, that's where calibration comes into play. If we want to calibrate concentration, we need to know something about the flow rate of the instrument. We need to calibrate it. And there are different ways that you might use standards to calibrate that. And once you do that calibration, then you can express the concentration in EVs per mil. And likewise, if we want to convert the arbitrary intensity of photons we detected in either our scatter or fluorescence channel, we need some standards to perform a calibration that lets us get to some size estimate, whether it's a diameter, a surface area, or a volume. Moving on to cargo, right? Again, we're only getting some number of photons measured on the instrument. And if we want to calibrate that in some meaningful units, whether it's the number of antibodies, the number of antigens, the number of fluorophores, there are standards and calibration protocols that can get us to those units. And so I think it's worthwhile thinking explicitly about what quantity are we trying to measure? What does the instrument measure? And then what sort of calibrators, both physical standards and protocols that will get us to calibration. And so you can see when you say calibration, you're really referring to a whole set of things that need to be considered. And I'll can leave that up there for a second, but I can take it around, take it off.
So John, this is really helpful, but so we have 15 minutes left, but just to move on the topic, so we've kind of described calibration. How about implementing it? So if users want to go away now and calibrate their cytometers and determine which are the best instrument, which are the best, what and the tools available and what's lacking.
So I think actually if you put John's slide back up, we can address it with each one.
Yeah, that's a good idea.
All right, so let me get back there. Hang on, I'm Zoom impaired. There we go. So I think probably the best place to start, because it's generally important for all cytometry, is with the fluorescence channels. Reagents and approaches to calibrate fluorescence in absolute units of MESF or ERF or antibodies per particle have been around for 20 plus years.
So let's get to the practical part. How often would you have to do this? I think for everybody coming in, like, is this a daily regime?
So I think that needs to be determined in your own. In our case, we will start daily, and once we figure out that there's insignificant variation over a week or a month, then we can move to weekly or monthly. And once we decide that there's better stability than that, we can push it out. I think you need to determine it empirically in your own lab.
So are the fluorescence standards, are they on the same par as your EV standards in terms of, dimness?
So there are newer ones coming out there. That has been the biggest limitation, I think, prior to now.
And I think that's one of the things that we've all talked about at different meetings and stuff, is the actual availability of getting these.
Yeah.
So we need to talk about this. I think this is a really good thing to address, the need for smaller, dimmer standards. So what I'd like to ask is what the utility of having necessarily the smaller, dimmer ones are. So our fluorescence scale should be linear, right? So we should be able to extrapolate based on what's available at the moment. So having smaller, dimmer standards is obviously useful if you have an incredibly sensitive instrument that doesn't have the dynamic range to detect the really big stuff. Yeah. But I don't think there's a need necessarily to wait for those before you start implementing these. If anything, it should just validate that the larger ones, extrapolations, were useful. So I don't know if everyone feels the same there.
But it's one of those things.
I think it depends on the linearity of the instrument down in the low end, and that's going to vary by instrument. I think some of the newer instruments are probably better, but if you're using an older instrument, I think there's a lot of non-linearity in the detectors down in the low end and the upper high end as well.
And if you don't have the standards there, you can't detect that.
Well, but in terms of, for the viewers here who are reagent developers, what is a better thing to work on first? Is it to have precise reagents, very consistent lot-to-lot variability, precisely measured each population, or is it to focus on dim particles or dim reference materials? And I would tend to think that for their workflow, it is probably easier to get very consistent materials, because if you've compared FITC MESF beads from one manufacturer to another manufacturer, you'll find that there are certain discrepancies potentially, right? So I mean, as a field moving towards development of materials, I would tend to say that it's better that we have precise materials rather than super dim ones for now. That's kind of like my own opinion.
It's not an opinion. And from a logical perspective, you definitely need both. Yeah. If you want to calibrate something according to the metrology handbook, you need to have reference materials in the signal range where you are measuring afterwards your particles. Else it's not a valid calibration. So you need well-characterized reference materials which are monodispersed for the property which you are measuring and have a known are related to SI units and have a signal range similar to your EVs for the property you're measuring. Only then it's a true calibration. So I think that's the ambition, but that's a long-term ambition. That will not be there in half a year or next year. I mean, this takes development, testing, and it doesn't only come with the reference material, but also with reference procedures. So if you have this material, how to use that? No.
So here's how I think about calibration and quantification practically. Edwin has some nice slides with a bullseye with the concentric red and white wings that he uses to illustrate accuracy and precision. You can also think about those rings as orders of magnitude of accuracy. And so in a perfect world, we'd like to get exactly the right answer, right?
Yeah.
In the real world, it's unlikely anyone's going to get any closer to within 20% of the right answer. Any of the numbers we're talking about here are going to be at best plus or minus 20% in my experience and understanding. Most of the methods that are reported, most of the measurements of vesicles are reported, frankly, aren't quantitative a factor of 10 or even a factor of 100, right? So we want to move the field from not knowing the right answer within a factor of 10 or 100 to something better. And, I think a factor of 2 would be a real big step forward. If we could all agree on our measurements we could compare our measurements within a factor of 2, we would have made great advances. And why do I pick factor of 2? Well, antibodies are bivalent, right? So you're never going to really be able to cleanly assess antigen abundance on a particle to better within a factor of 2 using antibodies, just because there's this uncertainty about whether the antibody's binding 1 or 2. So having long drawn out discussions or not calibrating because you can't get within a factor of 2, I think is not the right way to look at it. And so I would frankly advocate for using the existing commercially available standards to calibrate your results and reporting your results in terms of those commercially available standards, and not getting held up on the fact that 2 commercially available standards from different places give different results, because at that point we're sort of in factor of 2 territory. That's interesting questions to tease out, but we can't wait to solve those problems before we make additional progress. So I would argue for a little bit more pragmatic approach that says, let's at least getting people running the commercial beads. And one last editorial comment. I strongly believe that should be the responsibility of the core facility and not the user. I think that the core facility staff and personnel could do the field a great service by providing calibrated instruments to their users rather than just recommending that users calibrate for themselves. So some opinion there.
So yeah, I totally agree with you, John. And many of you know I run a flow core, and we do an article on a Cytoflex in our flow core. And I do run actually scatter calibration beads, and I have available MESF beads for purchase. So I pay for them out of our budget, and we expense it as consumables for users if they want to buy those beads. Simply because there is a cost involved, but it's not difficult to do. And with software academically free to academic users, it's actually not a difficult thing to do to calibrate. And there is a lot of protocols and published materials to teach people how to implement. But yeah, I agree that flow cores can definitely play a big part in the calibration process.
Because it's not just the beads, you need the expertise of how to do that.
Yeah.
And trying to train 50 users to do appropriate calibration is probably not the most efficient way to do it. I think having core personnel that know how to do it and actually do it for the customers is, I think, worth discussion.
Yeah, you mean to generate calibrated data for them?
Yeah.
They're paying $100 an hour for the instrument. I think they should get a calibrated instrument for that $100 an hour.
Okay, I'm going to jump in as my core director hat now, and tell you that there are many people out there that do not want you to do that, right? Because they don't really want to know the real thing.
Yep.
And they say, no, thank you. Please don't. That to me is a bigger challenge, right? I hear all the time, no, we've been doing it this way and we have all this data and we don't want, it's going to change that we do and we can't start over. You've all heard these kind of things. It's the same thing with cells, when you recommend certain protocols for good best practices for doing cells, right? People will challenge you because they've got all this data that's based on best practices, and trying to change that is very, very challenging. So I don't have the answer, but I think at least for people that are interested in producing good quality data, I'd work with them, but beware that a lot of people are just not going to be interested in this approach.
I think it's also what people see being published. And if all of the high impact EV journals are not showing calibrated data, as a core manager, it's really difficult to make that argument to the PI and say, hey, you should be doing it this way. And then they come back and they're like, well, I don't see this in any of the high impact journals. Why do we need to do this? And then there's an issue of trust, whether or not this is actually something that they should be doing.
I've taken John's approach, where if we're doing a collaboration with an academic person, we do the calibrations on our own. We supply that part of it. But to Joanne's point, I make sure now that if they're coming in for each of their probes, I'm like, okay, if you're going to do this, you need to do a titration. You need to show me that data. You need to show me that you understand that you're going to titrate, and do, even like if you're going to build a panel, do an FMO, and we'll help them with that. But it's because it's like, it's going to be coming out of our facility. I want to make sure that you're doing it correctly.
Yeah, but core managers are in a much more difficult position than that.
Yeah, no, I agree.
With the incentive that, you know, there's not, to Vera's point on the literature. So a lot of people are interested in ICEV and look to ICEV for the standardization of EVs. They trust the minimum guidelines for reporting their EVs. So the MyFlowSite EV framework is a position paper of ICEV, of ISAC and ISTH. And in there, it does recommend that fluorescence calibration be done regardless of your experiment. It should be something you should always do. So I think there are resources now and enough weight coming from these leading organizations that are saying, you know, this should be mandatory.
Yeah.
It's more convincing them to make it mandatory at peer review. And we can only do as good a job as the peer reviewers that are looking at those manuscripts.
Yeah, and I think that's true for people who are really interested in doing, there's several classes of people doing EVs, either who are really wanting to do it correctly and follow the Journal of Extracellular Vesicles, or ICEV members or ISAC members, but there's a whole lot of other people out there that are just Me Too's and jumping in and trying to do some EVs and are not interested. And then those papers are showing up in things like Cell and Nature because there's no reviewers on those boards that have a clue. So, you know, I think until, you know, this is a long time problem in cytometry, right? This is not new. It's applying to a new field. But until we get more of a consensus in the whole field, not people who are members of ICEV and read JEV or cytometry. And even cytometry, I have to tell you that there's sometimes some bad stuff in cytometry, which disappoints me. So, you know, I think we have a long way to go, but I think what we can do is take baby steps and educate at a time. You know, core directors should focus their attention on the people that seem to be interested in doing it, and we can hope that they will rise to the top and the others will then eventually follow.
I think that's we are running out of time now. If it's okay with everybody, we can extend for another couple of minutes to get some of the questions that are in the panel. But I think this has been a really great discussion, and it just echoes the theme of where we were at before, which is that we don't really have a defined way to analyze EVs, and hopefully everybody understands a little bit more as to why. There's a lot of things that still need to be decided on and considered, but there is actually, you know, we're starting with efforts, actually with a lot of people on this panel today, to forge a path forward. And there are guidelines out there for people to follow. And I think the take-home message is always be cognizant of the samples, the type of sample that you're running. Don't just go by, you know, what you read in literature. Always verify in your hands what they are, and calibration is a very important thing. We need to be speaking the same language for us to be able to compare our findings, and that's quite simply the reason why calibration is so important right now. Yeah, so before we end, a couple of minutes just to go through some of these chat questions. I don't know, Asia, if you can help me with some of them, because there's quite a list in here to go through. Does anybody have any questions they want to just ask, as opposed to me scrolling through all of the text that's just been going on here? If you want to just speak up.
I'll ask one because I'm always curious. If anybody has an internal control that they can run in any biological sample that's stable for hours and hours and hours, I would love to have it.
That's not a question, dude. That's you looking for it.
You're talking about a spike in?
I'm talking about a spike in.
Yep. So there's two ways to do controls. You can have an external control that a separate tube that you run along with your unknowns, or you can spike in, like what are some of the—
I want to spike in.
For cell analysis. True count beads you would spike in.
Yeah, but true count beads are going to be off the scale.
Right. And that brings up the other issue of, what effect does your bead have on your sample?
Yeah.
Which is
I finally found a bead that is stable in plasma for 200 consecutive runs, but it shows up right in the middle of my plot. So while it's stable, that's one check mark, but yeah, having it show up in the middle of your fluorescent plot is not good.
So I don't think actually we have the time to answer all of the questions here, and some of them are pretty long-ended questions as to what you would consider a small particle analyzer. What would you do to, what kind of markers would you have to identify, or common biomarkers for plasma E's? So these are all really big, meaty questions that we're not going to be able to catch. So we'll figure out some way to get these questions out to you. So we do have all of your contacts, and so if we want to answer something. For those of you who may not be aware, there is an EV Slack channel, flow cytometry Slack channel. Look them up, and a lot of the panelists that you see on here today are actually members of that Slack channel. So please do toss questions up there if that is what you wish answered. But yeah.
That's at evflowcytometry.org, and you'll find the link to that Slack channel.
I think all of us have answered questions on that one.
Yeah.
Okay. It sounds like you need a round 2 to get through the schedule.
I know we do. We need a small particles round 2. We'll get the Slack channel to host the next one. So yeah, it was great to have you guys all present. We ended up with over 100 people. And thank you, panelists, for sharing your time and your thoughts and your ideas. And it's promising. We're getting somewhere, and we're moving somewhere forward. We didn't get to touch on future directions, but I feel like the future is quite bright, and things to look forward to in the next couple of years.
Future's dim.
It's not dim. You have to say that
particles.
Okay, there you go. Give that one to John.
The future is bright with dim particles.
With dim particles. We'll hope we make the dim particles bright.
Yep.
All right. Thank you, everyone, again. And
thanks, Vera. Thanks for the committee.
Yep. Thank you.
We'll see you guys in 6 weeks on a different topic. Our next topic. What's our next topic? Does everybody know? Des, you're in charge of the next topic.
Yes, I do.
Instrument acquisition.
Yes.
All the details about instrument acquisition that you may want to know about.
So, and I think that comes back to the question that John has raised. Is there a best instrument?
No, that's no question.
No, but I think that's a general question that a lot of people ask. Well, how do you make a decision about buying a particular instrument, whether it's small particle flow, or microscopy? How do you make a decision to buy a particular instrument?
And right now we're limited by, the in-person demos are very difficult. So how do you what's the challenge, and how do you do this in a virtual way? So it should be a fun topic. We've got some fun panelists lined up for that one as well. Okay. And on that note, take care, guys. Stay safe.
bacteria have a volume 1,000 times less than mammalian cells, and we've accounted for that in the design of our system. As a result, we can detect populations unresolvable on conventional flow cytometers and changes in those populations as well.
Bio-Techne empowers researchers in life science and clinical diagnostics by providing quality reagents, instruments, custom manufacturing, testing services, and technical services. Our family of life science brands includes R&D Systems, Novus Biologicals, Tocris Bioscience, Protein Simple, and Advanced Cell Diagnostics. At Bio-Techne, science is our passion. It drives us to collaborate, develop, and manufacture innovative tools that help researchers from academia to industry achieve reproducible results.
The image stream is a high-throughput, high-data content imaging flow cytometer. We have recently released high-gain mode. Its superior photonic sensitivity makes it ideally suited for imaging small particles and extracellular vesicles.